So I kind of want to kick off our Q&A. And Dr. Lasokin, you can come back. Oh, perfect. With kind of a question. So, Dr. Lasokin, you kind of mentioned that you do an analysis basically of the patient's immune system and patients with immune systems that are weakened or exhausted, the T cells are exhausted, maybe worse. Um, should we use these immunotherapies earlier?
Dr. Patel, we're getting some feedback from your microphone.
So that's a great question. I think for sure these therapies, as as the initial late phase 1 trials are completed and a safe and effective dose is identified, and as sort of an initial kind of registration studies occur and things like that, concurrent with that, these treatments will be explored earlier on in disease natural history, including in patients earlier on in their treatment, like after 1 relapse or 2 relapses. And these kind of trials are already ongoing with idacel. There's a KarMMa trial, KarMMa 3. I can't keep track. 2? Yeah, I can't keep track of the KarMMa numbers. Yeah. Anyway, but it's a randomized trial of looking at idacel versus standard treatments in the relapse setting, which will determine what's the best approach. Similar types of studies are launching with bispecific agents as well.
Because as a patient, you kind of think, okay, I'm relapsing. Do I do another triplet combination? Do I go to a selinexor? Do I go to a melflufen? Do I try to bring, because what's causing the exhaustion, I guess, is the question. Is it the myeloma itself? Is it the treatment? So, I think we all are considering this chess game of myeloma as we think about it. So should I get into a clinical trial versus do the triple combinations or quad combinations? It's a lot for myeloma patients to think about, which is why we need people like you both.
Yeah, I mean, I think at any one point, and I saw some questions here in the Q&A asking specific sort of treatment-related questions. I think these are very challenging to answer in a broad sort of way because individuals are individuals. Individuals' myeloma is individual. The behavior is variable, and different approaches are appropriate for folks. And so I think there's always a question of how do you balance sort of the least amount of therapy, the best quality of life with the best longevity. And that's really the main focus. For sure, if a clinical trial is appropriate for an individual to answer that question or to explore a novel therapy, this is highly encouraged. This is how we advance the field. This is how we advance patient care.
Mm-hmm. And Dr. Patel, I want you to weigh in too. So I think when you're speaking, we're not getting the feedback. So go ahead and share.
Perfect. Yeah, so I completely agree. I think I'm biased and I tell my patients that's my conflict. I do clinical trials and my goal is to make treatments better, right, from what we have. And so I tell my patients that any clinical trial that we can get them on, I really try to get them on them. If the standard of care is an option, great. But if I think the trial could potentially offer something better, either outcomes or quality of life and toxicity, we have trials for all different types of things. But it's hard because there's so many things out there. And when you look at clinicaltrials.gov, for instance, it's very overwhelming to say what's important, what's not. And so I think you really need your doctor here to help you, right? And this is where having that specialist can tell you what seems to be the right thing to do for you versus, yeah, this probably is definitely not a good idea. We shouldn't go this route. The good thing is because we have all these options, there's more than one pathway. And so we can kind of hit a lot of the check boxes that people have in terms of what's important to them and in their life to make sure it's the right way we're going. But there's definitely some trials out there that they're not the right thing, at the stage you're at. And I think that's where you need help from us, to help guide and say, these are the ways you should go. But for most of the time, I will say clinical trials are always aimed at trying to improve what we can at standard of care. The problem with that is that there's only so many slots, right? So I don't want people to feel like I have to get onto that trial. I'm going to go to 50 different states to try to get on, because there's always going to be something next line that we can do. And it's really making sure you're connected through these programs to know what to look for, and having someone with you that can help you navigate that.
Mm-hmm. And what I would suggest is clinicaltrials.gov is sort of a nightmare, I think, for patients. So in HealthTree, we have, if you join the HealthTree Cure Hub at healthtree.org, you can enter in like your prior lines of therapy, your labs, and then in our partnership with Spark Cures, you'll be able to see all clinical trials you are actually eligible to join. So that will knock your list down from this 450 number to a more reasonable number. You can search by location or facility or type of therapy. So I would suggest that you go in and do that, because sometimes at smaller centers, especially, like if you're going to a local oncology center, they may not share the clinical trials that are open at a MD Anderson or at a Memorial Sloan Kettering. So you can do some of that homework on your own, and those tools are available for you. Okay, I'm going to start in with a question from Yuri. He says, many myeloma patients end up on dialysis or have kidney-related issues. Some doctors worry, and I don't know if this is the case or not, that bispecific antibodies might not be best for these types of patients because they're on lifelong immunosuppression. But is that the case? Is that a thing?
So, I mean, I think that's a great question. I don't think these are questions that haven't really been addressed by our current clinical trials, because they often will exclude individuals that are on dialysis or haven't, there's a renal function threshold for the trials. So can't answer it in a black and white sort of way, but I would say in theory, with the caveat that in theory and in practice is not always the same, that in theory, there's no reason really to think that a bispecific drug would not be effective in someone who has had either a kidney transplant or is in dialysis. But those are things that need to be explored with caution.
Mm-hmm. And Dr. Patel, you're welcome to jump in at any time. So.
Yeah, I will say I just had this conversation with one of my patients who's on dialysis, and we're trying to come up with the next treatment. And he wants to do CAR-T, and he'd be eligible in every other way except he's on dialysis. And so we've talked about, well, we wouldn't do the fludarabine because that part does go through the kidneys. But if we just did Cytoxan and CAR-T, could that work? But, obviously that's not the indication right now, and we'd have to really see if that's something we could do or not. But yes, this is what we push our pharma sponsors and doing trials in smaller groups of patients to prove that it's safe once it becomes standard of care, so then we can do it in everybody. But yeah, the original trials usually never, it's just certain patients that are on there, and then we have to prove that it's safe for all our patients so we can do it. So that's on us, and hopefully we'll be able to do that soon.
Mm-hmm. And I know the clinical trial, let's talk about the approvals for a second, because the idacel was just approved. The Janssen CAR-T is expected to be approved later this year. So when they get approved, they're only approved for the indication for the clinical, that the clinical trial was run under. So does that mean that if they were, is that true? And,
So I can start. So it's not necessarily fully what the clinical trial was run under, but what the FDA agrees based on the data that came out. So for idacel, for instance, it's 4 lines of therapy. You have to have had 4 lines of therapy and at least been triple exposed. And then you can get the drug. And so insurance will use that to say, are you eligible or not? And that's how we get it approved. But as Dr. Luskin said, there are trials right now, KarMMa-2 for high-risk patients in second line, and KarMMa-3, which is the phase 3 of this, like a confirmatory trial for this approval. that are earlier. So that's in lines 2 to 4. So the hope is that once we have that data, the FDA would look at it again and then hopefully approve it for sooner. So those are studies that are already going, so we wouldn't have to wait another 4 or 5 years. Hopefully it'll just be, a couple of years, maybe less, but it just depends on when we get that data. But yes, for right now, we have to go by what the label says.
Okay, and in the context of the kidney issue, like could patients with kidney issues get idacel or the Janssen CAR T after approval? I guess is my question.
Yeah, so it'll depend on, I think, what insurance allows and doesn't allow. I think we can try and go from there. If they say nope, then we wouldn't be able to. But, this is something that for us, for our department, we've actually come up with criteria that patients should at least have this, that we think it's still safe to do. So patients with ECOG of 0 or 1, that was what was allowed on the trials. We think ECOG of 2 is probably fine as long as it's related to the disease and someone doesn't have, heart failure or anything. But anybody with a lower, let's say, ejection fraction or cardiomyopathy, or maybe the kidney function GFR is less than 30, those are patients we would all bring up and discuss before we would even try to get the approvals. And then it would be if insurance approves it and we would be able to move forward. But yeah, those are all the things that I think a lot of centers are hoping to be able to branch out from what the trials really allowed.
I would stress that this was, FDA approved, was it March 25th or 6th?
Yeah, just barely, right?
Yeah. And so I think we'll probably have it available for patients or with the first leukapheresis sometime at mid to end of May. I think our approach is similar to Dr. Patel's here is that to be a little bit more conservative as we start out, to make sure that, our experience mirrors what was observed on the clinical trial, and to just get comfortable, if you will, with the product. Not to say that we haven't done plenty of CAR T cells. It's just to make sure that our experience really does mirror what we've seen on the clinical trial before we think about expanding. And I don't think it's not like we need to treat 100 people this way, but, 10.
Yeah, right. Okay, I have a question that's kind of related to Sharon's question. So Sharon's asking, can a patient do a stem cell transplant and then CAR T to extend remission, almost like a maintenance? At ASH, it seemed like I just got this sense that everybody was kind of chomping at the bit to maybe move some of these things up earlier and maybe replace transplant at some point. So how would you see that being, what strategy would you see being potentially best for something like that when it comes to transplant?
Yeah, so I think these are all things that are being actually actively explored. There are studies now designed for individuals with residual disease following initial therapy inclusive of transplant, for whom CAR T cells can be explored. There are also studies that are about to start, or maybe have already started, that are focused in on ultra high risk populations, and additional studies trying to explore CAR T as a part of first line therapy in those that would not consider stem cell transplant or would not be considered for stem cell transplant. So all these kind of, it's not exactly the head-to-head kind of question that you're getting at, but I think the sum total of this information derived from these types of clinical trials will help us understand this question a little to a greater degree, and ultimately it probably will be explored.
Agreed. I think, the question of T cell health, most of us from the lymphoma data, et cetera, we do know that the earlier we do it, your T cells are going to be better, and your myeloma is less resistant. That's with any therapy, we know that. To do it when your tumor burden is low, that's the big question. We do know that probably patients who have less tumor burden in general do better with immunotherapy. Your cells are able to control it a lot better. It's not 100%, but in general, it seems that, it's better to have some disease control going in. So if you have a transplant and you are MRD positive, so you have barely any myeloma there, what's going to happen to those T cells? Are they going to kill that and actually do better, or they won't expand? So those are really the questions that hopefully these current trials will answer for us. I have had patients on these trials and they've done great in terms of safety, but response, it's going to take time. Most patients do well post-transplant for years for a lot of patients. And so it's going to take time to really say clinically, did it change anything? But I do think that going sooner is probably going to be the best way. And if you look at lymphoma, it took them about 4 or 5 years, and they do have a trial that's now transplant in second relapse versus CAR T that they're trying to see if that is actually replaceable. So I think myeloma is probably heading down that road, but we just have to show safety first with all of these trials, and then in a few years we'll probably have that comparative trial.
Yeah, great. Sharon and Tom were kind of asking a similar question, like what's the median survival or progression-free survival for some of these CAR T trials, or even the bites? And these trials, I think everybody has to understand that they've been used in very highly relapsed patients with 6 to maybe sometimes 18 lines of therapy. So do you just kind of want to address that?
Sure. So this is exactly what I tell my patients, that I wish that some of our patients were cured from CAR T. That would have been great if we got that like lymphoma does. But I'll just say lymphoma is easier to treat. I'm just joking. I tease my lymphoma doctors all the time that myeloma, we've never been really able to get rid of it, right? So the fact that someone who's had 6 lines of therapy, they get 1 dose of cells at the 450 million with idacel, and their progression free is about 12 months, right? Which is a year, which is not forever. And I wish it was more than a year, but a year with no therapy, where my patients are going back to work and they're actually doing well. And besides the pandemic, enjoying time with their family, et cetera. I mean, that's huge. And so the question really is, how do we make that better? Going earlier is going to make that better, and we're going to get longer times. Also, the patients who got MRD or stringent CRs or MRD negative with the CAR T, they actually got 19 months or longer. So we know that those folks who have the deeper responses actually do better. So that's really the goal, is how do we get everyone to respond, and how do we get everyone to respond in a deeper fashion so that it improves those outcomes. And then one day actually cure some patients too. And that's the biology part that we really need to learn a lot about, the mechanisms of resistance and what else is happening around the rest of the microenvironment, so that we can tweak that to then make it less hospitable for that myeloma to come back.
Mm-hmm. And then, Dr. Lasokin, can you address also the idea of being MRD negative? I don't know what the data showed on some of these trials about if you're MRD negative, do you get longer progression-free survival in some of these studies?
So I think in general, the concept in myeloma appears to be that MRD negativity, right, and as well as sustained MRD negativity for a year or longer is associated with the best overall responses. So MRD is clearly a very important endpoint to achieve or to hope to achieve with therapy. Now, I often will see patients and they'll say to me, well, I'm MRD positive, but they're in remission. And so that leads to a lot of anxiety. And I would say that stable, low-level MRD positive disease also does quite well. And it's the dynamic changes that need to be addressed. Someone who's MRD negative becomes MRD positive. Those are the folks that are en route to something that, will require intervention, maybe not right away, but eventually. So I think it's not totally black and white, but for sure, MRD negativity seems to be a very important and good clinical outcome
Mm-hmm.
that results in more durable progression-free survival times.
Okay. Another question from Tim. He just says BCMA is seeming like a nuclear option. Do you want to address that idea?
Yeah. So I think a lot of our therapies are a nuclear option. In this kind of framework, myeloma therapy is not something to be taken lightly. It's something that, we have to address with care. It requires supportive care, anti-infective prophylaxis, thromboprophylaxis, and BCMA-directed therapy is not any different. For sure, you will. It's hard to say at this point whether or not our patients, are, because of the many treatments they've gotten prior to enrolling on these treatments or initiating these treatments, whether they enter the treatment already immunosuppressed or whether the BCMA is the cause of the immunosuppression or further resulting in immunosuppression. But for sure, low levels of immunoglobulins, folks that get Cytoxan and fludarabine as a conditioning regimen will have low T-cell numbers. And so these therapies do result in a fair bit of immune deficiency, and they do require careful attention to immune prophylaxis and infectious complications and rapid management of infectious complications.
On the CAR T, does that resolve? Like, you may be more prone to infection for a season, but does that take time? How long does that take to resolve? Do you have any thoughts?
Want to take that one?
Sure. So yeah, it's a multi-component, I guess, because it also causes neutropenia. And so for most patients, it takes about 2 months to completely resolve the neutropenia. And some people, it's even longer than that. So that already puts you at risk for infections. The T cell depletion, because of the risk of PCP pneumonia or PJP pneumonia, we have you on usually therapy for that prophylaxis for at least 6 months and sometimes up to a year. A lot of our patients will keep an eye on their CD4 counts or just sort of do a blanket 6 months. So I would say it's sort of like transplant where it does improve. The other thought though is, like in transplant, for some patients, their normal immunoglobulins start coming back up, 6 months to a year later, as their normal plasma cells are coming, but not the myeloma. And that's really what we're hoping to see. We don't have as much data with these patients with the hypogammaglobulinemia, but we give IVIG like crazy. So I give all my patients immunoglobulin so that it does bring it up. So it helps prophylax as well.
Mm-hmm. And Adrian is asking a question, and Dr. Patel, you talked about this a little bit. If you fail the BCMA CAR T, can you use another BCMA CAR T down the road? Interesting question, especially if you've used something else in the middle, like a bispecific or something.
Great question. And the biggest thing that all of us are looking at, I would say, I think there's been a lot of grants that have been thrown in this season for this exact question. So just like CD38, when we had DARA and all these new CD38s coming in, we really didn't know, can you retreat right after? Do you have to wait 6 months? I don't know if we still know the answer for most of those questions, but I think to have a different mode of getting to BCMA makes sense, especially if you have an ADC that you've used and now you're using a CAR T or a bispecific and then a CAR T. That the machinery that's going to what's going to kill that myeloma is different, right? You're just using the BCMA to get to the myeloma cell. The question really is what happens to that antigen. So if your resistance mechanism is that you lose BCMA, well, then you can't just give another drug that's going to go after BCMA because you're probably not going to get to that myeloma cell. But if the mechanism of resistance is not that, and you still have BCMA that's functional, that's there on the myeloma cell that we can still target, then it doesn't matter in timing, right? I think most people are going to say that we need to wait 3 to 6 months just to kind of give the best shot because we don't know the answer. But really, I don't know. I mean, we potentially could even bridge with something that's BCMA, knock the the myeloma down, and then give something different with like CAR T to really go after those myeloma cells. We don't know the answer to that, but I think a fantastic question and things that we are definitely looking at. I do think that using different antigens is probably okay, but when you knock one antigen down, what happens to the other one? We don't have the answer to that yet either for the majority of patients. So those are all things we need to learn.
Mm-hmm. Okay.
I would say
another question. Oh, go ahead.
Oh, I'm sorry. I was
No,
just
go
going to
ahead.
say that I mean, I agree with all of that. I think that there is data emerging, though, that, folks that have had BCMA-directed therapy will respond to GPRC5D-directed therapy or FCRH5-directed therapy. And even those that have had 1 or 2 prior BCMA-directed therapies will still respond to BCMA-directed bispecifics, for example. So I think there's a lot of open questions here. And I think it really does depend on what the mechanism of resistance is.
Yeah, right. Okay, let's talk about genetics in these immunotherapies. I know the whole idea of using an immunotherapy is to be kind of like genetic agnostic, really, just to have your immune system go after whatever is your type of myeloma, especially for CAR T, that's this manufactured product. Yeah, Dr. Lissow, can you mention that the 1Q gain patients might benefit from this FCRH? I always have to look at my notes because I can never, it's like FC something. FCRH5 bispecific antibody. Have any trials been looking at that in these early studies?
Yeah, that's a great question. I don't think, obviously we don't know the answer. It's too early to say, but for sure this will be looked at to see if one can correlate. It's sort of like a 11-14 for venetoclax kind of thing. It definitely begs that question. And I think it's just a matter of time before we have enough patients treated where there's enough number to actually ask that.
Because this is still phase 1, right? Or
yeah, exactly. It's sort of in the expansion phases of phase 1, not really in large phase 2 and not in phase 3 yet.
And more broadly, are you seeing any better impact for certain genetic features? Because some of these CAR T therapies have been in trials for several years now. Do you see any patterns or anything?
So I don't think that that's actually been looked at, as far as I'm aware. I don't know,
Sure.
if you have additional info on that.
Yeah, I think the only data out there is on Karma that was presented at ASH. They have some subset data where high-risk patients or people with high cytogenetic risk factors still had great response rates, about 60-70%, but their hazard ratio was a little bit lower, so less patients that responded. We don't really have PFS data on that. I think it really works for high-risk patients, but does it bring them to standard risk levels? It's probably not, but we don't have 100% that data. So that high risk still is high risk. That myeloma still figures it out, but this still has been some of the best responses, even in our high-risk patients, to this point that we've ever seen.
And a related genetics question by Anne Marie. She's just asking what genetics testing can be done on extramedullary myeloma and on nonsecretory myeloma.
Okay. Yeah. So, I mean, I think our, for sure, traditional cytogenetic analysis is very important, but also, especially with extramedullary disease, I think exploring sort of more targeted sequencing approaches for targetable lesions, things like BRAF mutations, KRAS mutations, can be helpful, as these are also targets for which therapeutics are available and can be explored.
Okay. And there's no, well, yeah, Elizabeth has a question about non-secretory patients because normally they're excluded from clinical trials. Is there any movement to include them, or no? It's just too difficult to track the myeloma.
So this is a pet peeve of mine. I don't know how others feel, but I certainly feel like this is an unfair exclusion. Lymphoma is non-secretory and you can follow it with imaging. Why can't you do that with myeloma? And there are reasonably good criteria in the IMWG for measuring disease by imaging, and both for response and progression with cross-sectional imaging. So for sure, the investigator-initiated trials that we start will all include non-secretory myeloma patients. Many of the sponsored trials, the registration trials, it's a much understandably more straightforward to determine response if one is measuring markers and % reduction in M spike or light chain or things like that. But So there's no reason, though, to think that that these agents would not work in non-secretory disease. So
Right. It's the same myeloma. You just can't track it in the blood. Yeah.
the other thing I would say is that soluble BCMA, as assays become available for that, clinical grade assays may be a place where there is another measurable component in the blood that can be tracked for response.
Okay, another question about what we talked about a little bit earlier. How do you measure, like a lot of these therapies are very expensive, and we may want to, know ahead of time if we're actually going to respond, if we're going to have side effects or cost associated with it. So is there some kind of T-cell panel or immune system panel? I know Dr. Chung at Memorial was kind of working on this idea, that we can have this panel run and say, okay, you're a great candidate for either the bispecifics or CAR T or something, and maybe you're not such a great candidate. Is there anything like that?
So I don't think there's anything like that that's sort of prime time quite yet, but I think for sure this is how we must proceed. There is emerging data that tells us that, certain T cell phenotypes are associated with better responses to CAR T and BiTEs. So one advantage of CAR T cell therapy is you can engineer your way out of T cell dysfunction by selecting the best T cells that you then subsequently manufacture your CAR Ts with. And those kind of studies are actually happening now. With bispecifics, you need to combine with drugs that help overcome whatever the endogenous T cell phenotype is. And that's a trickier, that's doable potentially, but that will require additional clinical study in patients. So these are things that I think all of us are thinking about, but they're not prime time.
Yeah, well, I'm glad you're thinking about them. Dr. Patel, do you have any comments on that idea also?
No, I completely agree. I think we need better biomarkers for everything. All our therapies, cost and toxicity wise. These are, big things that we're doing, changes. I have patients that respond so well to certain proteasome inhibitors, and then others who respond not at all to IMiDs, and I can't, really tell who that is from the beginning. And if in the future we can do this for all these therapies, that's the goal, right? It's going to take some time, but really with all the new data we're getting for old and new therapies in myeloma, that would really help us learn how to combine the different therapies and what sequence to put them in. Right now we use our best available knowledge, but I think we're subpar still right now.
Mm-hmm. Okay, Jane is asking a good question. Could soluble BCMA testing become the standard measure of residual disease, or does that not make sense? Because you can lose the BCMA but still maybe have myeloma. I don't know. Would it equalize heavy chain, light chain, non-secretory myeloma, or even replace bone marrow biopsy type testing?
I think soluble BCMA is really interesting. There are actually, as far as I'm aware, no actual sort of clinical grade assays that are routinely used to measure it, although we will hopefully have one soon. The challenge of measuring soluble BCMA will be that as you use BCMA-directed therapy, you have to consider competition and binding of soluble BCMA, and is that really indicative of disease burden reduction or just binding of your drug? BCMA loss is also an issue, as is increased gamma secretase activity or interactions with the ligands, because soluble BCMA can bind APRIL and can bind BAF, and so you can have complexes that will interfere with the assay. So it's actually quite complex. But I think, nevertheless, that taken in context as a whole, one looks at imaging, bone marrow result, light chain, M-spike, adding soluble BCMA to that context would be very reasonable, especially in an era where we have so many BCMA-directed therapies. I think it's a reasonable thing to explore and something that we're quite interested in exploring.
Okay. Anil was asking a question about something you talked about earlier, Dr. Lasokin, about this idea of myeloma stem cells. You know how you show they go through this progression of development. And I know one of the first studies was done, I think at the Mayo, no, maybe at UPenn, on CD19 with this idea that maybe these early precursor myeloma stem cells might have some CD19 on it. So when you think about these dual targets or just going after these cells that keep producing myeloma even after you're in remission, is that reasonable? Or is that something that's being investigated?
It's something that's being investigated actually by the UPenn group. There is some data that was reported by a group from China as well, I believe, with CD19 and BCMA directed CAR T. I don't recall that being earth-shattering, to be absolutely frank. It was interesting, but I think it's still an area that is being investigated, so more to come.
Yeah, I think there was only one patient that really responded
Mm-hmm.
in that way.
Mm-hmm. Correct.
Okay. Someone's asking, what's the recent experience with the BRAF or the mech inhibitors for patients that have that mutation? These are different targets, right? I mean, you both talked about this BCMA target as kind of the main target, and the other two as the bispecific targets. But what about these? I guess they're called point mutation targets.
So this is complex as well. So a MEK inhibitor has been studied in myeloma, really not very much efficacy as a single agent. BRAF inhibitors and the combination of BRAF and MEK inhibition in individuals with BRAF mutations can be quite active in some patients. Exactly who and why, not clear. I would say that RAS-RAF mutations are present in probably 50% of myeloma patients. It's a very common mutation, but it's the challenge is disease heterogeneity. And if one can eliminate or target, 30% of the disease that has the mutation or 40% of the disease that has the mutation, the heterogeneity somehow suppresses the benefit there. And so that's been, I think, one of the challenges. These areas are still being investigated and studied based on sequencing, directed therapies. So there's several studies that are out there like that. There's the MyDrug study sponsored by the MMRF. And we also have a study in collaboration with MGH open at our center, focused specifically on KRAS and BRAF mutations.
Mm-hmm. Dr. Patel, do you have any comments on that idea?
Same thing. We've had a couple of patients that, had extramedullary disease and had increase of some of these, and we've been able to successfully treat with a targeted therapy. But for most patients, it's not the only issue. And so it really does then mutate more, and you have to find different therapies. So I think at least somewhere to start, but we still have a ways to go.
Sounds like the immune system would be the better. I don't know. I asked one of the myeloma doctors at one point, like, what's the best ultimate cure for myeloma? And the answer was like getting your immune system to recognize it and kill it. And because, there's so many different mutations, it seems like it'd be really tough to try to target just one with a specific inhibitor. Okay, Sam is asking, in what way does having had an allo transplant, stem cell transplant, to affect the relevance of using a CAR T? Not that many in myeloma have had allo, but some have.
Yeah, that's a fantastic question. I think, unfortunately, most of the trials at the beginning didn't allow for patients who had allo because we didn't know what would happen with CRS and neurotoxicity, especially if your cells are still your donor cells. Could that increase GVH? All these questions. I have had a few patients that have been able to go on to either bispecific or CAR T now. Some of them do allow for prior allo. It's still a few studies. And, handful of patients we haven't seen more toxicity in, but I think something we'll have to definitely still keep a close eye on, because your immune system is very different once you've had an allo transplant. But we also have allo CAR Ts, and so it's sort of they're different in terms of using an allo CAR T when you have your own immune system versus using your donor to make CAR T. But I think if your disease is overall controllable, making your T cell specific for myeloma makes sense, as long as you don't worsen GVHD, et cetera, which can happen with allo patients. But yeah, there's very few patients. It's kind of like extramedullary disease, or non-secretory disease, that most trials, plasma cell leukemia, non-secretory and allo transplants are usually not included from the trials, but a few are starting to let them on, which will give us more information.
Okay, I want to ask one final question before we end. What do you see as the best possible potential strategy for maintaining response? Because like you said, these responses are really unbelievable. I mean, in myeloma care, totally unbelievable to have patients on these prior lines of therapy respond at 95% response rates. But maintaining durability, what do you both, and I'd like you both to weigh in on this, what do you both see coming that would be the most effective to maintain that amazing response?
So I can go first so that Dr. Lisso can have the last word. I honestly, I am with CAR T specifically, I think the chemo-free period is so big. And so I don't want to completely get rid of that. I do think some type of consolidation, maybe with maintenance of, let's say, an IMiD or a cell mod, probably will be worth it to really build your bone marrow after you've had those CAR Ts and give them the best shot at persisting. And then potentially giving a chemo-free period. And let's say someone's MRD negative and they become positive, can I give another cell dose at that time? This is all, there's a lot to learn still about antibodies, et cetera, but can I give another cell dose, same antigen, different antigen, or can I give a bispecific at that point? My goal would still be to have some periods where you have chemo-free with CAR T, but just really being able to monitor it much closer. And when we see it start coming up, give something else, another cell dose or a different therapy, rather than waiting till someone's relapsing. And if there are CAR T still there, I would give something different to help those CAR T proliferate again, versus if there's no CAR T there, I would do maybe another CAR T infusion.
Okay. Dr. Lisokin, what are your thoughts?
So I think that as we proceed, I actually think that combination therapies, combination IO therapies are probably the way that things will progress. This concept of having a GPRC5D CAR T cell, in the preclinical model, where you have heterogeneity and you have some cells express BCMA, some don't, but maybe those will express GPRC5D. And this is just an example. So if you sequence those therapies as CAR T and give them together, or you sequence 2 BiTEs, or you sequence CAR T and BiTE, or you just give them all at the same time, right? I mean, wouldn't that be cool? I mean, it's sort of like total immunotherapy.
Right.
Right. But I think we're a ways away from all these things because, one has to proceed with caution.
Yeah.
You can't overstimulate the immune system. It gets exhausted, if you will. I mean, that's in the parlance that we use of exhausted T cells. And chemotherapy, traditional treatments like IMiDs and carfilzomib are very effective, and people do quite well with them and tolerate them quite well. And so I wouldn't throw these things out. So I think at the end of the day, we have a toolbox. We can try to use the kitchen sink approach or try to, think carefully about what's the best tool for the job. And I think as we proceed, additional information will emerge that will help us. Things like 1114 and venetoclax, for example, right? Mm-hmm. That is, I think, a paradigm that we need to think about. Like, What's expressed? Which IO should we use? What do the T cells look like? Can we use BiTEs? Should we use CARs? Should we sequence them? Should we, go back to using, immune checkpoint blockade, something that's been complicated in myeloma because it doesn't really work on its own. But maybe in the context of these therapies, or in the context of, terminally exhausted T cells, that is something that can be investigated. So there's sort of, it's a very hopeful time, I think, from the myeloma patient perspective. It's an incredibly exciting time from a myeloma clinician perspective, because there's just all these amazing tools. But it's also humbling, because we're not curing our patients. We have all these tools. We suck as tool users. We need to get better. Like, what's going on here? So, I think, rationally designed combinations, that's the way to go.
Mm-hmm. Well, I agree that I think the learning will be what to do in the right order for which patient and how to combine it, and that's the challenge and the exciting opportunity too, right?
Mm-hmm.
So I just want to thank you both for joining us today. It's been amazing having you. I think it's unique also having those that are both myeloma experts and immunologists, because those two sometimes don't always go together. So thank you so much for spending your time with us on a Saturday. We really appreciate you.
Thanks so much for having us. It's been a pleasure.
Thank you.